Hello students, welcome to LEELA'S tutorial. So in last class we have discussed about plant tissue culture, where you are making use of plant cells or tissues in order to produce a genetically modified plants by using your tissue culture technique. So today let us see what are different types of plant tissue cultures. Okay, first it can be based on the media you are using, and it can be based on the explant you are using. If you are using the same chemically defined synthetic media with solidifying agents such as agar-agar or agarose, you call it as semi-solid cultures. Or if you are using the same chemically defined synthetic media or the natural media in a liquid form, then you call it as suspension cultures. Right? So in case of semi-solid cultures, the explant will be inoculated over the surface of the semi-solid media. Right? You will be embedding it to the half over the surface of the media. Whereas here in case of suspension cultures, it will be freely floating in the liquid. So here you can see this is the semi-solid media where the explant is over the surface. If it is a liquid media, here inside the liquid, the explant will be suspended and cultured. Is that clear? So next, coming to the types of types of tissue culture based on the explant. So whatever the explant we are using for tissue culture, based on that the name of the cultures are defined here. So first one, it is the callus culture. So what is callus? Here you can see a undifferentiated mass of cells which are unorganized, right? These are unorganized, undifferentiated, dividing cells. So here, why they are dividing? Because when you inoculate any plant cell in a tissue culture media, because of the totipotency, those cells have an ability to divide and get transformed into a complete plantlet. Right? Based on that, you get a callus. So now if you are going to culture these callus in tissue culture media to produce a plantlet, then you call it as callus culture. Right? Next, coming to the organ culture. So here any part of the plant can be taken here. The plant organs are the shoot system, root system, leaves. Right? So from those organs, plant organs, you are going to produce a plantlet. That is referred as organ culture. Next, coming to the seed culture. So here in the seed culture, you are going to derive the explant from the plants in vitro. Right? So here it is a natural seeds which you are going to take. Before that you are going to sterilize them, surface sterilize them properly. Then you are going to go for your culture process. Next, coming to the protoplast culture. What do you mean by protoplast? If it is a plant cell, suppose this is a plant cell, right? This is the cell wall. Here you have all the internal organs of the plant cell, the cytoplasm, nucleus, everything. So here, this is the cell wall. The plant cell devoid of the cell wall is called as your protoplast. Only the inner one. Right? So here this plant cell, when the cell wall is removed, it consists of all the cell organelles present in it. Right? Embedded in it. And these protoplasts are used for culturing process. So in order to produce your somatic hybrids, you are making use of this protoplast culture. Right? By making protoplast fusion, you can produce those hybrids and hybrids. Then next is embryo culture. So here again, in the embryo, it can be a mature embryo which you can take from the fertilized seed. Otherwise, immature embryo from the seed which fails to germinate. So these explants can be taken to produce a plantlet. This is referred as embryo culture. Next, coming to the pollen culture. So here the male gametes are the pollen grains, right? So these pollen grains are used to culture and produce a haploid plants. See here, in all these cases, these all are somatic cells, the diploid number of chromosomes are present, right? Whereas here you are taking only the male gamete, that is the pollen grains, in order to produce haploid plants. In another case also, here instead of taking the pollen, in case if the anther is very minute, you can't take the, or you can't isolate the pollen, you can't separate the pollen if the anther is very minute. So in that case what you will do? Directly you will take the anther and culture it. Right? So in case of pollen culture and the anther culture, you are taking only the male gametes so that you can produce haploid plantlets. Right? And the last one is single cell culture. So once you are taking any tissue, if you are mashing it and again suspending it in a liquid, so what happens? All the intact cells will be on the surface, right? When you gently centrifuge them, you can get a single cell suspension, right? So these single cells which are suspended can be inoculated into the liquid media and can be grown as suspension cultures. These are mainly used to produce secondary metabolites. So all the techniques we will discuss in detail in the further classes, right? So here let us see, once you are selecting the explant, you are selecting the explant and selecting the type of culture you have to do. Either it is a semi-solid or it is an suspension culture. You are going to select based on the explant used, right? So let us see what are the steps involved in plant tissue culture. The first step is selection of the explant. What part or which part of the plant you are going to culture, you are going to select. Right? After selecting any one of the explant, the next step will be inoculation and incubation. Once you select the explant, you are going to perform surface sterilization process for the explant. Then after preparing the media, you are going to sterilize the media in autoclave in case if it is heat stable compounds. In case of heat labile compounds, you make use of filter sterilization. Right? So after sterilization, the explants are inoculated into the media and incubated under sterilized conditions, the environmental conditions where you are going to maintain the suitable pH, temperature, light intensity and all. Right? Then it is the initiation. As we told, the plant cells have totipotency. That means it is having ability to develop into a plantlet. That means it has to start dividing. So here it is the initiation process where it is supplemented with all the nutrients and the minerals required for its growth. Next one is the multiplication. So once the growth is initiated, it multiplies rapidly to form a callus. It is the multiplication. Once the callus is formed, again here you are going to provide them with the growth hormones such as cytokinins and auxins. So here auxins is to cytokinin ratio where is 1:3 and 3:1 based on the tissue or the based on the shoot formation or the root formation. Right? If the auxins are rich, then it results in root formation. If the cytokinins are high, then it will result in shoot formation. Right? So after multiplication from the callus, you are allowing them to produce shoot system, then followed by the root system, or root system followed by the shoot system. So once the complete plantlet is formed, right? When you are calling it a plantlet, when it is having a complete fully developed plant which is having all the organs, the shoot system and the root system, now these plantlets are sent to greenhouse. So in the greenhouse, it is the acclimatization process where the plantlets are getting adjusted to the new environment. Right? So in the greenhouse, you are going to provide all the nutrients and the climatic conditions required for those plantlets to grow. Then after acclimatization, that is the plantlets are getting adjusted to the new environment. Here then it is you are shifting them to the nurseries where you are going to maintain the natural environment. Right? So in this way you are going to produce the plantlets by using different types of tissue culture media and also different types of explants. Is that clear? If you have any doubts, Put it in comments.